So our study is regarding the use of whole genome sequencing to develop patient-specific minimal residual disease assays for pediatric solid tumors. So we've been working with neuroblastoma and Ewing sarcoma samples in particular. And we are focused on expanding the use of quantitative molecular monitoring, which is pioneered in the management of acute lymphoblastic leukemia, to pediatric solid tumors. So the sensitive measurements of minimal residual disease are actually central to contemporary risk stratification and treatment allocation algorithms for patients with acute leukemia. However, the measurement of minimal residual disease in pediatric solid tumors is predominantly undertaken in the context of clinical trials and is not currently used for prognosis or risk stratification or even for treatment allocation. In the past, limiting factor in developing solid tumor MRD has been the difficulty and the cost of being able to identify patients and tumor-specific targets that can be used for MRD detection. Many of the currently used techniques for molecular monitoring in solid tumors have relied on the expression of tumor-specific genes. For example, in neuroblastoma, there is a panel of genes including tyrosine hydroxylase, FOXP2B, DCX, etc., or methylated forms of certain genes. But with the increasing availability and reducing cost of whole genome sequencing, we have shown the feasibility of using whole genome sequencing data to develop patient-specific minimal residual disease assays, which are individualized tumor-specific assays across pediatric cancers, including neuroblastoma, Ewing tumor, and acute lymphoblastic leukemia. So just to summarize the study, our study shows that whole genome sequencing data can be used to identify individualized tumor-specific DNA breakpoints and can be used as reliable targets for PCR-based MRD analysis in pediatric cancers. And all these assays are optimized based on the Euro MRD guidelines, which was actually devised for ALL. And these patient-specific assays show high concordance between two different assay platforms, which is quantitative PCR and droplet digital PCR platform. And we are able to quantitate one tumor cell in 100,000 normal cell, or in the range of, like, say, one tumor cell in 10,000 to 100,000 normal cells. And we have shown that in serially collected patient samples, which are samples derived from bone marrow or from peripheral blood, we're able to track the disease course by targeting these specific DNA lesions as MRD targets. Basically, this is something which has never been tested and optimized on solid pediatric tumors. And we are following the ALL approach and the guidelines set for ALL to transform these assays in solid pediatric cancers as well.